It's a pleasure to be here. My name is Alex Pasteur. I'm a partner with F Prime Capital based in London, where we have a team of 4 investing professionals. My other colleagues are based in Boston and San Francisco. So the total F Prime group is about 20 investing professionals. And we're now on our 6th venture fund, which is a $550 million fund. Most of my involvement in the cell therapy for solid tumor space comes from our investee companies, Adaptimmune, where we participated in the Series A crossover financing back in 2015, and some other companies like Therapeutics in the US, and Precision Biosciences as well. So that's me. I'm moderator, but I'll now ask Oz and Kim to introduce themselves. Oz.
Hi, good afternoon, everybody. Thanks for coming this afternoon. Azam, President and CEO of Tmunity. We're a Philadelphia-based T-cell engineering company with both CAR and TCR platforms, and pretty much with a focus on solid tumors and difficult-to-treat liquid tumors such as acute myeloid leukemia. We've been around since 2015, and a small company of about 50 individuals now, completed a Series A raise in early 18, and are in the clinic right now with 2 programs with ambitions to have 2 more programs in the clinic by late this year and early next year.
Great, thanks Alex and Oz. So Kimberly Freeman, I'm at Adaptimmune. We are a leader in developing T-cell receptor T-cell therapies, and primarily in solid tumors. At this stage, we did have a asset that we've licensed to GSK that did focus in some hematologic malignancies. However, at this stage, we're focused in over 10 solid tumors. We're based in Philadelphia and Oxford, UK. In Oxford, we have most of our research staff and other corporate functions. In the US, we have our CMC manufacturing headquarters, as well as other clinical roles. And at this stage, we have 3 clinical assets, 2 cancer testis antigens, MAGE-A4 and MAGE-A10, and alpha-fetoprotein targeting hepatocellular carcinoma. And it's really been a long road at Adaptimmune in terms of developing these agents. But I think what we found, and I heard so frequently this morning, is many companies referring to themselves as fully integrated. And I think it's one thing we take very seriously at Adaptimmune, not only from a development and research perspective, clinical development as well, but having our own in-house manufacturing now has allowed for drastic improvements in how we bring these therapies to patients. So great to be here.
Thanks, Kim. So I thought we'd start the discussion by focusing on targeting, and maybe making a very crude 30,000-foot view summary of the T-cell therapy field, to observe that in leukemia, we've seen some absolutely spectacular data. In lymphoma, some pretty good data. But in solid tumors, so far, it's more of a question mark. And in terms of asking why might that be, the first question that came to my mind was the question of targets, and whether or not there is a lack of good targets to really get the approach to work in solid tumor, or indeed whether we're going to need to go to some second-gen approaches to unlock targeting, and that might involve targeting multiple antigens, using logic gates or veto signals, or perhaps opening up the universe of eligible targets by incorporating suicide switches and thereby sailing perhaps a bit closer to the wind than we might have wished to a few years ago. So that was my first question, and I'll throw that to Oz to comment.
So all of the above, right, when you think about it for solid tumors. But the way we try and think about it within Tmunity is that antigenicity is important. But we try and do it in a 3-pronged approach. So we think about optimizing the target, optimizing signaling for the T cells, and overcoming immunosuppression. There's probably about 5, 6 other things we think about, but if I had to sort of crystallize the 3 areas that we really focus on, it's probably those 3 areas. And I think then when it comes to solid tumor land thinking in particular, is it going to be about multivalency? So I personally believe certain tumor types you're only going to tackle and potentially get responses if you take a multivalent approach. An example would be glioblastoma. There's enough evidence now in the field that no one single target's going to address that kind of disease state. But there may be signatures out there for certain cancers. I mean, we know certainly for certain rare diseases and certain rare types of cancers, for example, we're looking at an antigen specifically for medullary thyroid cancer. We have a target called GFR alpha 4 that's, we think, only uniquely expressed in that population. Now, you could think that in theory you could have a CD19 approach there where a single antigen can do it, providing you get the right affinity tuning done of your SCFE complex, providing you get the co-stimulation the right way, and you're able to power these cells on to stay on sufficiently. So I think it really is going to be very dependent on the tumor biology, and I think our understanding of the microenvironment has now exploded in the past 5 years. It continues to do that. I mean, there's not a week goes by when you don't see a new state-of-the-art paper coming out in terms of how you think about the microenvironment. But I do think that certain solid tumors, it is going to have to be a multivalency approach, and not just multivalency, but then how do you deliver those cells in? So for glioblastomas, I mean, clearly intratumoral deliveries, where are you going to deliver that? How are you going to deliver that? But for certain cancer types, there could be a shot on goal with a single monovalent approach. But let's see, it's still early days, right, in terms of thinking about that, certainly how we're thinking about the field, but when it comes to targeting specifically in antigens.
Got it. And Kim, with engineered T-cell receptors, perhaps you have an embarrassment of riches when it comes to targets. Is that playing out in the way that you might have hoped?
I think we are seeing things play out. I think in terms of our targeting with specifically the cancer testis antigens, we're in a broad range of tumor types. I think we've seen with sarcoma that we can get enough of a single antigen approach in that disease state to have efficacy. I think what we're seeing is hints of activity. We had a poster at ESMO last year that showed in an ovarian cancer patient, we had 27% regression in her target lesions. Now, that said, I think what the hurdle is the heterogeneity of all these multiple tumor types and the heterogeneity of each of the patient's starting material, the prior therapies they've seen, the different modalities they've seen really impacts the outcome of these therapies. And so whether or not the single antigen or the right target approach will hold true for all of the solid tumors we're exploring, I think we have yet to see that. That said, we do take a second-gen approach very seriously because we have a very robust translational program that takes not only tumor specimen but also blood specimens at set times through a patient's course post the T-cell infusion. And that information is vital to guide us to, as I said, those specific nuances we may see with specific tumor types and the bar that we have to cross to get the efficacy we need.
That's a nice segue to my next question, actually, which was really around strategies to overcome immune evasion and strategies to modulate the tumor microenvironment. And obviously, there's a number of strategies that are being developed in the industry. Foremost among them, perhaps, programming of intrinsic checkpoint blockade, IL-2, TGF-beta blockade, etc. As you think about modulating the tumor microenvironment in your respective companies, I'm interested in your views as to which types of these strategies you think is most promising. Maybe Oz to start with.
So I think it goes back to really understanding your tumor biology really well, because it's really dependent on that, right? So you touched on two that we're involved with. So TGF-beta. TGF-beta is overexpressed in the tumor microenvironment of adenocarcinomas, so prostate cancer, pancreatic cancer, potentially lung cancer as well. And so it made sense for us when we were looking at our prostate program, which is our lead program, in which we built on the shoulders of others and realized targeting the antigen alone may not be successful. So for example, in the case of prostate cancer, we're targeting PSMA, prostate-specific membrane antigen. And that actually trying to understand what makes T cells fatigued in that environment and prevents them doing the job, how can you harness that? And we were fortunate to have a team around us of really, really good synthetic biologists in the form of Carl and his team at Penn and another founder, Yangbing Zhao. And they figured out a way to actually mutate the receptor that binds TGF-beta with a view to actually preventing TGF-beta stimulation at the cellular level. So that's one way we've looked at it, but that's particular for that microenvironment. And then the other approach we've taken is with CRISPR. So we have been successful with the team at Penn to actually knock out 3 things in a cell. And so, how much can you actually do to a cell? That was one of the first feasibility things we were trying to figure out. And it's remarkable how much you can do to a T cell and it still be actually potent enough to do something, at least in vitro and in vivo. So we were able to knock out the alpha and beta chains of the normal T cell receptor, and also knock out PD-1 and introduce a new transgenic receptor targeting NY-ESO1 like colleagues at Adaptimmune, different transgenic TCR, but same principle. And we started dosing patients in that trial earlier this year at Penn, so it's the first attempt at a PD-1 intrinsic knockout. We don't know if that's going to work. Early days yet, at least let's establish the safety of CRISPR. But hopefully that gives you a flavor of how we're thinking about it, and there could be other checkpoints and other modalities to knock out if you're looking at specifically immunosuppression. But that's just one piece of the puzzle, right? Again, it goes back to how we think about, you can knock out the checkpoint, but is the cell still going to be stimulated enough to be able to move forward? So, one of the other approaches, in tangent a little bit with your thinking around suicide switches, et cetera, is can you actually have a CD28 PD-1 switch, for example, where on the tumor surface you actually are getting the PD-1 ligand to be triggered, which causes a co-stimulation with CD28 to make those cells overcome resistance. That's a different approach than knocking PD-1 out, for example. Mm-hmm. So I think we've got a lot of armamentarium and tools in the synthetic biology toolbox now. The question is getting these into clinic and validating them, and sort of that's what companies like ours and many here are trying to do, but hopefully that gives you a bit of a flavor of how we're thinking about it.
It does. And Kim, how's Adaptimmune thinking about the TME and some of your second-gen approaches?
Yeah, absolutely. Second generation is very important to us, and we have a number of ongoing programs, TGF-beta, phosphodiesterase, we have CD8 alpha, a number of approaches that we're looking at. We will be filing an IND later this year in an unnamed second-gen approach, and I think that it's exactly like Oz said, it's the translational data that will guide us and lead us to the optimal situation, and it may be different dependent on tumor type, as you were saying as well, Oz, with prostate cancer. I think for us as well, though, the combination approach is not one we should ignore, and there's a lot we'll need to learn with our NY-ESO program in multiple myeloma. We did see an upregulation of PD-1 in patients that were progressing on T-cell therapy, and so a logical next step would be to combine with an anti-PD-1 therapy. So for us, it's as vital as our first-gen program, but I think, like I said, we'll see success in various tumors with the first-gen, and we'll likely need a second-gen approach in others. With our AFP asset, that's in very early stages. We're in dose escalation right now. We just got through 100 million transduced cells. We're at the 1 billion cell dose, but at 100 million cells, where we really were just simply exploring safety, we did see necrotic tissue in T-cell infiltrated tumor for that patient, and those data were just reported out at AACR this past month in Atlanta. So I highly recommend, if you want to see the poster, you can look at that on our website. But certainly for us, it's a priority to keep going. I don't think any company stops with first generation, and we'd be remiss if we didn't explore all these options.
Now, it's interesting you mentioned combinations. I'm interested in your view of the end game here, maybe sort of 10 or 20 years out. And, if you imagine a world where you need a couple of cytokines in there, a couple of checkpoint inhibitors, it's 3-4 mechanisms working at the same time, which maybe that's what we need. Is that going to be delivered by a single cell doing everything in a very, in an elegant way, or are we going to need, multiple administrations of different drugs to get there, which, seems more complicated, but maybe at the individual product level is simpler. So I'm just interested in your your view was, as someone in the trenches thinking about this every day, as to where you think it ends up a decade from now?
Boy, that's a profound question, isn't it? So I think, we're thinking heavily about payload delivery that's meaningful in the microenvironment. So clearly, interleukin payloads are going to be key. How that's done, is that done through constitutive elements, or are you looking at exogenous approaches there? That's a conundrum we're still trying to figure out. I think the other area that's attracted and garnered a lot of attention is combinations with oncolytic viruses, hot cold tumors, that concept as well. And we're still at that base level of foundation, right? Because no one, to my knowledge, has got to that stage where they've taken a TCR or CAR effectively into clinic with a combination strategy yet. It's probably going to be the next ushering of Gen 2, Gen 3 products. But I think then the fundamental question is, are you really looking at a master cell bank allogeneic approach in which you can program all these elements? And I think eventually that's where the world is going to get to. I mean, we're in essence dealing with a math and engineering problem, I think, when it comes to allogeneic versus autologous, personally. I think, we've got the tools and the technologies now to know we can knock out so many things, but can we maintain viability? Is that going to be through a master cell bank, a universal cell, or what have you? And I think that's going to determine how your payload delivery is ultimately going to happen. It may probably happen in the autologous setting before an allogeneic setting, is my own personal belief, but maybe I have more of a bias there towards an autologous versus an allogeneic platform there. But 20 years from now, I mean, you would like to fast forward and think that our understanding of tumor biology has got to a point where we can really decipher at a genomic level very efficiently and quickly what payload's going to matter, right? Mm-hmm. What's going to work for one tumor type versus another. And there's a whole cadre of upstream master regulators that I don't think we've really fully appreciated. I mean, I'll just take TGF-beta as an example. I didn't really think about it until we really started to dive in, how much of a master regulator something like TGF-beta is. And there's probably a whole host of things that we probably need to dust off the shelves and look at again. And new regulators are being discovered all the time in terms of those approaches that we can switch, turn on, turn off. So I think it's going to be a painful track, right? It's not going to be done overnight. It probably is going to take 10 to 20 years, to be honest. But I think the fundamentals in terms of synthetic biology are all coming together nicely. And it's who can combine these modalities in a safe way, in a benefit-risk way that gets regulators comfortable. How do you think about the CMC components of this? Because the more things you pack in, the more complex your CMC elements become. And so how regulators now start to think about these combinatorial approaches, because they are going to come, and they're probably going to come sooner than we think. Is the framework in place yet for that? I'm not so sure. So every time you say you have a base product and then you add in an element, right? Is that a new IND each time? Are you looking, how are you going to figure this out, right, and speeding up that process? So some general ramblings from my part on how I think this could evolve.
Yeah, I'll just add that I think if I look at it from a practical perspective, so Oz gave a great scientific rationale for why I think this will take time, but I think even just from a safety perspective, right, we know that timing of a PD-1, can't be perhaps directly with a T-cell receptor product. And so figuring out the science and the safety of how these different targeted effects impact the patient cell, off-target toxicities, and then not to mention cost of goods of combining these things and how you do that and develop them from a manufacturing perspective, I think are just things that will also take time and cannot happen overnight. But it's something that I'm hopeful for, and I think the data will teach us a lot. And I think all these companies in the room are doing great things to get us there.
Yeah, it's interesting to me that you've both mentioned TGF-beta as a thesis that your respective companies believe in, and I'm aware of another one, a strong company in London called Autolus, and they're also going after TGF-beta. Yeah. So that's 3 companies, and I'm sure if I looked, I'd find lots of other TGF-beta. To what extent can you learn from each other? So if someone's in the clinic a couple of years ahead of you with their second-gen cell, with the, which has bolted in the TGF-beta blockade, if it works, great. Is that de-risking for your own program? If it doesn't work, does that help you would that mean you deprioritize your own program? or frankly, is it all going to come down to the individual construct, and it's hard to tease out lessons from other companies?
Oh, I think it's hugely beneficial to learn from others. I mean, if you look at the TGF-beta field, I mean, TGF-beta isn't new. There's a graveyard full of products that were TGF-beta blockers that were toxic because people couldn't figure out that, TGF-beta is a protein complex that's required for normal physiology, and in the attempt to block it, people ended up killing patients, and that sort of car parked the field. So we learned a tremendous amount when we just look at companies that were in the TGF blockade space and thinking about how do you block TGF-beta in a T cell versus systemic administration. So I think you always look and inform, and we have been looking at other companies and their approaches. We think we stumbled upon, a unique way to look at it within a T cell. But you're constantly evergreening and thinking and learning from each other. Will we get there? Hopefully. If not, it's going to help inform the next generation. And maybe it's not TGF-beta. Maybe it is, as I mentioned before, and we already have plans for this, in our prostate program, a CD28 PD-1 switch, where that might be as potent and deliver what needs to be done. Mm-hmm. Could you combine that with a TGF-beta component? Probably. Should you? Depends on safety, right? You may end up producing something that's very potent and uncontrollable. So, yeah, always learn from others. And I think that's the beauty of this sector and this field, right? We are always pollinating and learning from each other in such real time. I mean, it's amazing when you think of where the field went from 2012 with a handful of patients that were initial CD19 proof of concepts to where the field is now. And it's only going to get better, I think, providing we're willing to share and learn. And I certainly feel in this sector more than others, we really are willing and wanting to collaborate with others more.
I mean, I've never seen this amount of collaboration. I've been in oncology research for 25 years, and I think that, whether it was monoclonal antibodies or targeted tyrosine kinase inhibitors, whatever the pathway was, there was never this amount of collaboration, and I completely agree. We felt with our AFP platform, once the Eureka data came out in liver cancer, that that was showing activity and that this was a viable target for us. That was hugely reassuring, and definitely gave us a little more optimism in progressing the program through the safety dose escalation stage, because alpha-fetoprotein is expressed in the liver, and you don't want to damage the healthy tissue of the liver. And so I completely agree that we have to learn from each other and share the information to best move these programs forward.
And Kim, I mean, Adaptimmune is one of the first companies to take multiple T-cell therapies for solid tumors into clinical translation, following the great success in the hematological cancers. Just having followed the story for a few years, one observation I might throw out there is that, a blessing in the liquid tumor setting was that the hematologists were already familiar with bone marrow transplant and conditioning and harsh conditioning regimens. And so that it sort of the approach fitted seamlessly with clinical standard of care. But in solid tumor cancer, you're often dealing with surgeons who are not used to handling immune cell therapies, let alone harsh conditioning regimens. So to what extent do you think that has been holding back translation in the solid tumor setting? And is that now, are we starting to make headway now in terms of educating and finding the right physicians to do the trials with who really are up for this?
Yeah, so I'll answer it in two parts. I think that the first is absolutely there's been a learning curve for solid tumor treaters with lymphodepletion regimen. I mean, cyclophosphamide and fludarabine are not common agents in these disease settings. And so I think the flip side's been that we go to centers of excellence where the CAR T therapies oftentimes are approved because of their knowledge and awareness of the side effects that come along with these therapies. You need this robust infrastructure that these academic centers have to really successfully conduct your trials. So it's sort of two parts to this story. There's definitely a challenge in educating them. I have some physicians who say, I'm horrified of that lymphodepletion regimen, and they're very nervous about potential side effects to the patient. But once they have that experience at the clinic level in that setting with that specific tumor type, they're much more comfortable to proceed and enroll more patients. I do think the next wave beyond the academic center, specifically in solid tumors, is the community referral in the US. And even in parts of Europe, referral from approved treatment centers from hospitals who perhaps don't have access to these therapies. And so this is something we have to take a very close eye to in terms of commercialization and clinical development of these assets, is to make sure that the education and awareness is there, and also that the data to go along with it that says you can do this and do it safely. So I think it's a very important point that you raise, Alex.
Mm-hmm. Yeah, I think just to add to that, I think it's what you're seeing in our space, which is very refreshing to see, is that even in phase 1 settings, multi-center trials are going to become the norm. So, we've all grown up with, phase 1 small molecule biologic trials, typically doing in a PK/PD environment, phase 1 CRO type of unit. But that's not going to be the case for cell therapies and gene therapies. Certain gene therapies, for sure, where it's monogenic disease and a handful of centers, that maybe you could see one center being super specialist and receiving those patients, that kind of makes sense. But if you look at solid tumors, for example, you need to be able to, I think companies have a duty of care as well to think about how do you educate investigators as well, who really want to get into the space but have immuno-oncology experience, but not necessarily TCR or CAR-T experience. Mm-hmm. And they're hungry to do that. So a lot of our trials, our phase 1 trials right now, are multi-center trials. In that, we start with dosing certain trials at Penn. Certain trials that we're about to kick off, actually, we're not going to be starting at Penn, we're going to be starting in other centers. So I think that model's changing, and there's an appetite, certainly by those that are immuno-oncology versant, that they are prepared now to sort of really build up the infrastructure around them to handle the benefit-risk challenges that arise with these therapies, mainly cytokine release, but others as well. And so I think you're going to see, and that needs to happen because the volume of companies that are trying to push through INDs, that there needs to be some given the system to start to treat more patients, certainly within the US setting.
Very good. So almost 3 minutes left. I'd love to ask you both, what's the single most exciting thing you're aware of in the T-cell therapy for solid tumor field? Kim.
Jeez. I think for me, the fact that we are starting to see anti-tumor effect in solid tumors. And so it may not be the ideal level that we need to perhaps overcome a regulatory hurdle in certain disease states, but the fact is we are seeing, hints of activity and in first generation, and I think that's very important, and it's taken a lot to get to this point at Adaptimmune. It's almost 2 decades worth of work, and so I think we all learned that this is not something for the faint at heart. It's going to take work, but I think that at Adaptimmune specifically, the translational body of research that we're generating is immensely important to the field, much less our own assets. And so for me, it's the most exciting time I've ever had in my career to be part of this cell therapy and bringing these assets to patients. I'll just end with, we brought a sarcoma patient in to our facility a year ago now, 40-year-old with her 3 kids, and she came in and she's just one of those amazing success stories. We saw this morning with one of the corporate presentations, the patient story, and we can't lose sight of that. And she's been disease-free now for about 3 years. And what she spoke about resonated with me because as a mom of 3 kids myself and not battling what she's certainly going through, she said the debilitating side effects she experienced from all the other therapies she'd received had just left her with no quality of life. And the cell therapy, a one-time infusion, she had some myelosuppression and other things from the lymphodepletion, but the fact that she was one and done, she said, and she really was living life, back to work, taking care of her kids. They toured Philadelphia that week, and for me, there's nothing better. So,
thanks. Oz?
So I think it's really exciting when you start to see signals in man. We're really good at curing tumors in rats and mice. And I think we've crossed that line now. And I'm really optimistic in the next 4 to 5 years, you're going to see initial data that's compelling. Whether we can then reproduce it and make it persistent and durable is the real challenge. I don't think there's going to be any shortage of signal. We're already seeing that. So I think we're on the cusp of something quite brilliant in medicine in the next couple of years when it comes to solid tumor thinning. Not for all solid tumors, but I think there are certain tumor types that we really have a shot on goal here right now to have a good run at least initial responses and potentially taking a run at cures as well.
Thanks, Oz, and thank you, Kim. And that brings us to the end of our half hour.
Thank you,
Alex.
Thank you.
Thank you.
Here's a few.